Hi everyone, my name is Kayla Schaefer and I will be covering Digestion 2 with you all. Before we dive right in, let's recap the digestive organs. These include the mouth, the pharynx, the esophagus, the stomach, the small intestine, the large intestine, the liver, the gallbladder, and the pancreas. In order to remember these organs, I have come up with an acronym to make it a bit easier. It goes, My pet Elmo sees Simon like a legit golf pro. Not only is it important to remember these organs, but it's important to remember their functions as well. I have broken down the organs and their major functions into this chart to have another visual at remembering these functions. The mouth is the beginning of chemical and mechanical digestion. The pharynx is where swallowing occurs and prevents food from entering the nasal cavity and the trachea. The esophagus connects the pharynx to the stomach and peristalsis is occurring. The stomach is where the breakdown of food is occurring, and mechanical and chemical digestion is occurring as well. The small intestine is the major site of nutrient absorption. The large intestine is where water and salt is absorbed and chyme is turned into feces. For the liver, bile aids in digestion and absorption. The gallbladder stores bile, and the pancreas helps with enzymes that aid in digestion and absorption. Let's first begin by looking at an overview of the anatomy of some of the accessory organs such as the liver, the pancreas, and the gallbladder. The liver is the largest gland in the body and is also one of the most important organs. The liver can be divided into two primary lobes, which are the large right lobe and the significantly smaller left lobe. The porta hepatis, also known as the gate to the liver, is where the hepatic artery and the hepatic portal vein enter the liver. The hepatic artery delivers oxygenated blood from the heart to the liver, while the hepatic portal vein delivers deoxygenated blood and nutrients absorbed from the small intestine to the liver. The liver is also organized into hexagonal structures called lobules. Each lobule consists of three main components, which are the hepatocytes, the hepatic sinoids, and the bile caniculi. The hepatocytes are the liver's main cell type. They are responsible for filtering blood to remove nutrients for storage and are also the cells that produce bile. Within each lobule, hepatocytes stack up to form tall columns. In between these columns are channels called hepatic sinoids. They carry blood from outside of the lobule towards the central vein in the center of the lobule. Between adjacent hepatocytes, there are small ducts called bile canaliculi. They accumulate bile produced from the hepatocytes. Bile then flows first in the bile ductules and then into the bile ducts. These bile ducts form together and exit the liver as the common hepatic duct. The pancreas is nestled into the curvature of the duodenum. This gland has both exocrine and endocrine functions. Exocrine functions mean it is secreting digestive enzymes, while endocrine functions mean it releases hormones into the blood. The exocrine part of the pancreas is organized into cell clusters called acini. These acinar cells secrete enzyme-rich pancreatic juice into small merging ducts that form two major ducts. The larger duct fuses with the common bile duct before entering the duodenum. The smaller pancreatic duct, also called the accessory duct, runs from the pancreas directly into the duodenum. Scattered throughout the exocrine acini are small endocrine cells called the islets of Langerhans. These cells produce hormones such as insulin and glucagon, which control glucose levels in the blood. The gallbladder is nestled in the shallow area on the backside of the right lobe of the liver. It stores and propels bile into the duodenum via the common bile duct. The gallbladder can be divided into three regions: the fundus, the neck, and the cystic duct. The fundus is the widest portion of the gallbladder and narrows to become the neck. The neck angles superiorly as it nears the hepatic duct, and the cystic duct bridges the neck and the hepatic duct. Surprisingly, there is no submucosa in the gallbladder wall. The wall's middle area is made up of smooth muscle fibers, and when they contract, the gallbladder's contents are pushed through the cystic duct and into the bile duct. Now let's dive into the rest of the material. We will now begin looking at chemical and mechanical digestion. Mechanical digestion is the process of breaking down foods into digestible particles. On the other hand, chemical digestion is the process by which foods are broken down into smaller substances that are easily absorbed in the body. Mechanical digestion mainly occurs in the mouth, while chemical digestion mainly occurs in the stomach. Mechanical digestion is mainly due to the functions of the teeth, while chemical digestion occurs due to the function of enzymes. In the mouth, mechanical digestion includes chewing and swallowing, while the chemical digestion of carbohydrates and fats begin. In the stomach, mechanical digestion involves the peristaltic mixing of chyme with stomach contents. Chemical digestion of proteins and fats is occurring. In the small intestine, mechanical digestion is occurring due to segmentation, while there is further chemical digestion of carbohydrates, fats, polypeptides, and nucleic acids. In the large intestine, there is mechanical digestion occurring due to segmentation and mixing, while the only chemical digestion occurring is by bacteria. Now that we have an overview of what chemical and mechanical digestion is, let's begin to look at the primary secretions that occur in chemical digestion. Chemical digestion starts in the mouth, where enzymes salivary amylase and lingual lipase are secreted to begin the breakdown of carbohydrates and fats. In the stomach, secretory cells aid in chemical digestion. These secretory cells are parietal cells, chief cells, mucous neck cells, and enteroendocrine cells. Parietal cells produce hydrochloric acid and intrinsic factors. Chief cells secrete pepsinogen, which is the inactive form of pepsin. Mucous neck cells secrete acidic mucus to coat the stomach lining and protect against stomach acids. Lastly, the enteroendocrine cells release the hormones gastrin and histamine. These hormones help to stimulate further release of hydrochloric acid. In the pancreas, there are several secretions that help with chemical digestion. Pancreatic protease helps digest proteins. Pancreatic amylase digests starches. Pancreatic lipase digests fats. And pancreatic nucleus digests nucleic acids. Now let's move on to discuss the phases of gastric secretion. There are 3 phases in gastric secretion, which are the cephalic phase, the gastric phase, and the intestinal phase. These secretions are all under the control of hormones and the enteric nervous system, which is also referred to as the second brain. The first phase, the cephalic phase, takes place before food even enters the stomach. The smell, sight, or even thought of food triggers this phase. When you see a piece of food, impulses from receptors in your taste buds or in the nose are related to your brain, which return signals that increase gastric secretion to prepare your stomach for digestion. The second part, the gastric phase, lasts approximately 3 to 4 hours and is triggered by neural and hormonal mechanisms that were initiated by the entry of food into the stomach. When the food enters the stomach, it activates stretch receptors, which stimulate an increased secretion of gastric juices. Partially digested proteins and rising pH levels stimulate the release of histamine and gastrin from the enteroendocrine cells, which also generates parietal cells to increase the production of hydrochloric acid. Also, the release of gastrin activates smooth muscle contractions, which allow for mechanical digestion and transport the chyme into the duodenum. In the final step, the intestinal phase, it has both excitatory and inhibitory elements. Partially digested food fills in the duodenum, which causes intestinal mucosal cells to release a hormone called intestinal gastrin, which further increases gastric juice secretion. At the same time, secretin and cholestokinin are secreted into the duodenum, which triggers the release of pancreatic juices and bile. The gastric inhibiting peptide restricts the functions of the stomach, which causes the pyloric sphincter to close, blocking additional chyme from entering the duodenum until it is empty again and ready to receive more chyme. In gastric secretion, specific hormones are released such as gastrin, cholecytokinin, secretin, histamine, and the gastrin-inhibiting peptide. I previously mentioned some of their functions in the previous slide, but now we will focus specifically on their actions. Gastrin is a stimulatory hormone. Endoendocrine cells trigger its release, and gastrin acts on both parietal cells and chief cells. Gastrin stimulates parietal cells to release hydrochloric acid and stimulates chief cells to release pepsinogen. Histamine is also triggered by enteroendocrine cells and only acts on parietal cells, which promotes further release of hydrochloric acid. Both of the actions of gastrin and histamine decrease the pH levels in the stomach. Secretin, cholecytokinin, and gastric inhibitory peptide are hormones produced by the duodenum. Secretin stimulates the secretion of bicarbonate and digestive enzymes by the pancreas. Cholecytokinin stimulates the release of bile from the liver and the gallbladder. And the gastric inhibiting peptide inhibits gastric secretion, slows gastric emptying, and motility. These hormones are secreted by specialized epithelial cells located in the mucosal epithelium of the small intestine, and they then enter the bloodstream to reach their target organs. To conclude, the digestive system and its accessory organs are a complex network of organs that work in unison. The accessory organs are vital to the overall function of the digestive system, each with its unique role in the process. Mechanical and chemical digestion are means of digesting food and its contents and occur in different organs in the digestive system. During these processes, there are different secretions and hormones released, all with their own important functions. With that being said, I hope you have learned how these processes and organs work and that you enjoyed my presentation. Thanks for watching.